Frontal lobe neurobiology, imaging, and criminal patterns, integrating prefrontal dysfunction with forensic phenotypes. Abstract. This article synthesizes evidence linking prefrontal cortex, PFC, structure, function, and connectivity to criminal patterns. It outlines key PFC subregions, neuroimaging methods, recurring imaging signatures found in offender samples, behavioral phenotypes that emerge from prefrontal dysfunction, and the practical, clinical, and legal implications of using frontal lobe imaging in forensic contexts. The piece concludes with ethical cautions and research recommendations for more responsible, translational work. Introduction. Scope: explain why the prefrontal cortex matters for behavior relevant to criminality, decision-making, impulse control, affect regulation, moral judgment, and social cognition. Aim: provide an integrated framework linking neurobiology, imaging markers, and observable criminal patterns so clinicians, researchers, and legal stakeholders can interpret findings with nuance and restraint. Framing principle: brain differences are probabilistic contributors to patterns of behavior, interacting with environment, development, substance use, trauma, and social context. Prefrontal neurobiology and imaging methods. Key prefrontal subregions and functions. Ventromedial PFC, VMPFC, value representation, affective integration, moral reasoning. Orbitofrontal cortex, OFC, reward evaluation, reversal learning, behavioral flexibility. Dorsolateral PFC, DLPFC, working memory, planning, cognitive control. Anterior cingulate cortex, ACC, conflict monitoring, error signaling, motivated control. Common neuroimaging tools and what they measure. Structural MRI, cortical thickness, gray matter volume, detects neurodevelopmental or degenerative differences. Diffusion MRI, DTI, white matter microstructure, indexes frontolimbic and frontostriatal connectivity. Functional MRI, task-based, region-specific activation during inhibition, moral judgment, or empathy tasks. Resting state fMRI, intrinsic network connectivity, highlights dysregulated PFC limbic coupling. PET and SBECT, metabolic and neurotransmitter markers, e.g. dopaminergic or serotonergic signals, when available. Lesion and neuromodulation studies, causal inferences from focal injury or stimulation. Imaging findings, criminal patterns, and behavioral phenotypes. Recurring imaging signatures. Reduced ventral/medial gray matter and cortical thinning in VMPFC/OFC correlate with impaired moral judgment, callousness, and diminished affective empathy. DLPFC hypofunction or reduced volume often co-occurs with poor executive control, planning deficits, and impulsive, opportunistic offending. Altered ACC activity links to impaired error processing and impaired adaptation following negative feedback, increasing repeated risky behaviors. Disrupted frontolimbic connectivity, weakened top-down control over amygdala, aligns with reactive, affective aggression and difficulty regulating anger. White matter abnormalities in frontostriatal tracts correlate with disinhibition and poor delay discounting, relevant to substance-linked crimes and impulsive assault. Behavioral phenotypes map to neural patterns. Impulsive reactive offender, diminished DLPFC/ACC control plus, weakened frontolimbic connectivity, right arrow impulsive assaults, domestic violence, crimes under intoxication. Instrumental/callous offender, VMPFC/OFC deficits plus intact cognitive control but poor affective integration, right arrow premeditated exploitation, deception, and low empathic response. Executive dysfunction offender, widespread PFC thinning and frontostriatal disorganization, right arrow chronic antisocial behavior, difficulty following rules, frequent recidivism. Illustrative vignettes, anonymized, composite. Case: a early adolescent with childhood maltreatment, DLPFC thinning, and poor inhibitory activation on stop signal task. Pattern: impulsive property offenses escalating with substance use. Case B, adult with focal VMPFC lesion after TBI, preserved cognitive IQ but marked reduction in guilt and poor social conduct, leading to exploitation and repeated boundary violations. Implications for practice, policy, and ethics. Clinical and rehabilitative implications. Targeted interventions, cognitive control training, emotion regulation therapies, and pharmacologic strategies aimed at neuromodulatory systems can be matched to the PFC profile. Risk management. Neuroimaging contributes one data point among many: behavioral history, psychosocial risk factors, substance use, trauma for individualized risk formulation. Rehabilitation monitoring. Functional and connectivity measures may serve as biomarkers to track therapeutic response when used longitudinally and ethically. Forensic and legal considerations. Limits of inference. Group-level imaging effects do not translate into deterministic individual predictions. Caution against over-reliance in courts. Admissibility and interpretation. Imaging should be accompanied by expert testimony clarifying probabilistic meaning, alternative explanations, developmental, environmental, and measurement limitations. Bias and fairness. Socioeconomic and racial confounds, comorbid substance use, and prior trauma can produce brain differences unrelated to moral culpability. Equal application of standards is essential. Ethical guardrails. Avoid deterministic narratives that remove agency or stigmatize. Protect privacy of neural data and consent for any forensic use. Prevent misuse for discrimination or neuroprofiling of disadvantaged groups. Promote transparency about the uncertainty and limits of neurobiological evidence. Future directions and recommendations. Longitudinal, developmentally informed cohorts that combine neuroimaging, genetics, detailed environmental histories, and repeated behavioral assessments to parse causality. Multimodal integration. Joint analysis of structure, connectivity, task activation, and neurochemistry to capture network-level dysfunction. Intervention trials with imaging outcomes to test whether changes in PFC function predict reductions in offending or recidivism. Standardized forensic guidelines co-developed by neuroscientists, ethicists, clinicians, legal professionals, and community representatives. Equity-oriented research designs that explicitly model socioeconomic, racial, and trauma-related confounds, and avoid pathologizing social disadvantage. Conclusion. Prefrontal imaging enriches our understanding of neurobiological contributions to criminal patterns, but must be integrated with behavioral, social, and developmental data. Responsible translation requires probabilistic interpretation, ethical safeguards, and a focus on rehabilitative rather than deterministic narratives. When used cautiously, frontal lobe neurobiology can inform tailored interventions, refine risk formulations, and encourage policies that address both neural and social drivers of harmful behavior.